Thanks so much for joining me for part 3 of Parker Biology, Cell Communication. In this edition, we'll be continuing our discussion of reception, transduction, and response. We mentioned in our last video that receptors can be found either on the plasma membrane for hydrophilic types of signaling molecules that don't move easily through, or receptors can be found intracellularly for small hydrophobic substances that can pass through the membrane. Now we're going to look more in depth at 3 main types of receptors. We'll look at G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. First up, we'll examine the G protein coupled receptors. These are receptors that, go figure, are coupled to G proteins. Notice that the G proteins can take on 2 different forms. They can be inactive when they're bound to GDP, but they can also become activated. So when the signaling molecule binds to the receptor actually undergoes a conformational change. The receptor changes shape and it binds to the G protein. At this point, GDP is displaced by GTP, the higher energy form. And at this point, the G protein can be called activated. From here, the activated G protein leaves the receptor, comes over and binds to an enzyme, and at this point, the enzyme is activated. Once it's done activating the enzyme, the G protein returns to the lower energy inactive form. It cleaves a phosphate group, and that GTP is now GDP. So overall, we see this constant shuttling. When a signaling molecule shows up, we'll see the G protein come over to get activated, shuttle back across the membrane to activate an enzyme, and then it goes back to the low energy form. I'm going to jot down a few notes over here on the margin just to remind me that we're looking at these different forms of the G protein, and once it's activated, it can go on to activate enzymes. So it's interesting enough to see how binding of the ligand can bring about these changes in the G protein, which in turn can activate an enzyme. But to me, it's even more interesting to note that up to 60% of medicines that we use influence G protein pathways. So again, our understanding of these receptors can really help us improve our treatment of disease. The next type of receptor we're going to look at is called a receptor tyrosine kinase. Here we can see that without signaling molecules present, the receptors are actually individual polypeptides. We can see these are separate chains. However, when the signaling molecule or ligands bind, the receptors dimerize. These 2 subunits come together to form a dimer. This activates tyrosine kinase, which adds a phosphate from ATP to each of the tyrosines on the tail. Notice here we have tyrosines, but because tyrosine kinase has been activated, we can now see phosphate groups from ATP being put on to each of those tyrosines. If there's 6 tyrosines, how many ATPs will it cost to add a phosphate group to each one? Very good, 6 ATPs, 1 phosphate from each being added to those tyrosine tails. From here, the receptor proteins are activated, which goes on to activate relay proteins, ultimately bringing about cellular responses. So we're still looking at this idea of reception, transduction, a series of changes, and ultimately the cell does something about the message it receives, the response. Again, I'm going to take a moment to jot down some main ideas from this, but hopefully you can see that it is the presence of that signaling molecule, or molecules I should say, that brings about the dimerization, which then brings about phosphorylation, transduction, and cellular response. The next question wants us to clarify what a kinase is. So the ASE ending gives me a pretty good clue. I know that this is going to be an enzyme. And I can see that it's activated in this region. And the activation allows for the phosphorylation. And that's exactly what a kinase does. It's an enzyme that transfers a phosphate group to a protein. Just like we saw cyclin-dependent kinases playing a role of phosphorylation to help push cells through the cell cycle, we can also see these tyrosine kinases bringing about phosphorylation. The third type of receptor is known as an ion channel receptor. And I'll give you a big clue, these involve ion channels. As we can see in the picture, the gate is closed until the ligand or signaling molecule binds. Once that ligand binds, the gate opens, allowing certain ions to pass through. Once that signaling molecule is released, the gate again closes. I'm going to jot some notes over here to remind us about this idea that ion channel receptors only open when the appropriate signaling molecule is present. In summary, if you see ions moving as a result of a signaling molecule, you're looking at ion channel receptors. If you see receptors dimerizing as a result of signaling molecules, you're looking at receptor tyrosine kinases. And if you see a G protein shuttling between a receptor and an enzyme, going between its inactive and active forms, then you're looking at G protein coupled receptors. All of these are great options for cells to use to take in a signaling molecule. No matter which type of receptor the cell uses, the next step in cell communication is transduction. So when the receptors for the signaling molecules are located in the plasma membrane, the transduction stage is usually going to be a multi-step pathway leading to specific responses. What's nice about this is it allows for amplification of the signal. If the molecules activate larger numbers of molecules at the next step, meaning it's not just one molecule talks to one molecule talks to one molecule. This could be 1 talks to 10 talks to 100, so we can see the message getting much louder throughout the cell. Also, having multi-steps within this transduction pathway allows for coordination and regulation. The cell can finely tune what kinds of responses it carries out. Most of the time, the original ligand or signaling molecule doesn't actually enter the cell. Instead, the message is passed through a pathway in a different form, oftentimes through conformational or shape changes in the proteins, and this can be caused by phosphorylation. Remember that it is a kinase that helps transfer phosphate groups from ATPs to proteins. So we said that transduction is typically a series of changes, and here we can see a whole series of changes in the form of a phosphorylation cascade. And what this means is that a bunch of different molecules are going to be getting phosphorylated and then help phosphorylate the next, which will then help phosphorylate the next. So this is a cascade because we see this trickle down effect. Do you think that all of these proteins can stay phosphorylated forever? No, they clearly need to go back to their inactive form so that this message can be sent another time. So in order to dephosphorylate, there are other enzymes called protein phosphatases. And these help catalyze the removal of those phosphate groups, which make them inactive and therefore available for reuse. Take a moment and pause this video, please, and label anywhere on this diagram where you see the activity of protein phosphatases. Label it P, P, wherever you see dephosphorylation. I see this active version becoming inactive by the release of a phosphate group, So I'm going to show that the enzyme protein phosphatase is at work there. Also along this dotted line, and also along this dotted line. Everywhere that those phosphate groups are being removed, making these active proteins now inactive. In this instance, we have a lot of different protein components, but not all parts of a signal transduction pathway always have to be proteins. Let's flip it over and see. Even though proteins can be a big part of transduction pathways, sometimes we see small, non-protein, water-soluble molecules, and we can refer to these as second messengers. These can also help with that transduction stage, spreading messages through the cell. Two great examples of these non-protein components are cyclic AMP and calcium ions. These aren't proteins, but they do help spread these messages through the cell. In fact, we can notice that ATP is actually converted into this cyclic AMP, second messenger. Which enzyme is catalyzing that reaction? Good, adenylyl cyclase. So while proteins and secondary messengers both play the same role in a signal transduction pathway, the relay molecules, or proteins, are typically large and they don't really move easily through the cell. Second messengers, on the other hand, are nice and small, they're water soluble, and they can diffuse quickly. Next, please pause the video as you read through this scenario and answer the question. Pause the video now, please. What did you think? When a ligand's released, what prevents all the cells in the body from being affected? Very good. It comes back to that specificity of the receptor. Only certain receptors fit each ligand. So we've examined reception, transduction, and now we'll flip to the last page to look at response. The response is what the cell is actually going to do based on the message it's received. And this response can take place in the nucleus or in the cytoplasm. A great example of a nuclear response would be if a signaling pathway functions as transcription factors. This will help determine which genes are transcribed and ultimately translated. So this is determining which proteins the cell is going to make. And that's an important message that cells can get. Make this protein or stop making this protein. Cytoplasmic response can be brought about when activities of the proteins in the cytoplasm are regulated via signal transduction. Here we bring back a familiar example, epinephrine. We see that epinephrine is going to bind using a G protein linked receptor. Following reception, here's transduction. And remember we said that these transduction pathways can result in amplification. And here we see how. Notice that each enzyme in the pathway can act on many molecules of the substance. It's not just one molecule. We're looking at exponentially more molecules, ultimately leading to this response where glycogen is broken into glucose. It's through this cascading transduction pathway, though, that the message gets louder. One signaling molecule brings about a whole bunch of cellular response. Finally, we've got an instance where highly regulated cell communication is really important, and this is called apoptosis. Cells that are infected, damaged, or have reached the end of their functional lifespan do need to enter a programmed cell death. They've run their course, and they do need to be broken down through apoptosis. So different cellular agents help break down their DNA, help break down their organelles, and in effect recycle some of their parts. This is where cell communication and regulation is so important because we sure wouldn't want cells to accidentally undergo apoptosis. Here we see normal formation of a vertebrate limb, relying on that apoptosis to form individual digits. Thanks so much for joining me for part 3 of cell communication.